Welcome to the third topic of Module 5, Hair Examinations. We will begin by discussing hair and explaining how hair grows. We will also discuss the various portions of the hair and how they are used to show differences between human hair and animal hair. We will then discuss the various microscopic characteristics that are used for human hair examinations in a forensic laboratory. And finally, we will discuss hair evidence as it's collected and packaged. Hair is all around us and can be found in a wide variety of criminal investigations. The average person loses approximately 100 head hairs a day. So exactly what is a hair? A hair is one of the numerous fine and cylindrical filaments that grows from the skin in a hair follicle. The follicles themselves are initially created by going through several stages. Part of these stages create the actual hair canal, which develops into the hair follicle, with the dermal papilla being at the base of the canal itself. The dermal papilla is the portion of the hair follicle from which the hair actually grows. The follicle also has a muscle attached to it, a pilia erector muscle, and this is the thing that causes your hair to stand up when you get chilled. There's also a sebaceous gland that provides moisture and oil to the hair. Once that hair follicle has developed, hairs can start growing in the follicle itself. This is a continuous cycle beginning with the anagen phase. In this phase, the hair is actively growing. So the root portion of the hair begins, and then further as it grows, developing the shaft. The hair grows, and it's composed of a protein called keratin. Eventually, it will emerge through the skin and continue to grow in that anagen phase. At some point in time, the hair will transition into the catagen phase and then finally into a telogen phase where it's in more of a resting stage. You can see from the examples on this slide, in the anagen phase, that hair is beginning to grow. In the catagen phase, you can see the root of the hair still down in that dermal papilla. But as it goes into the telogen phase, that hair is being pushed out of the hair follicle, and eventually it will exit the follicle itself through normal shedding. At that point, another hair will begin to develop in that same follicle and once again start over in that anagen phase. So it's a continuous cycle. There are several portions of the hair that the analyst is involved in when they're doing their examination. They'll look at the root of the hair, then the main portion of the hair shaft, but then also the tip end or the distal end of the hair. In the main portion of the hair shaft, they always look a little bit closer for three different portions of the hair. The cuticle, which is the outer layer of the hair, the cortex, which is the main body of the hair, and the medulla, which is the center core of the hair. The cuticle is a very thin, generally transparent layer that surrounds the hair shaft. It's composed of scales, and they overlap in a wide variety of different patterns. The patterns can be used to help distinguish animal hair from human hair. Human hair will always have an imbricate scale pattern. The cortex is the main portion of the hair. This portion of the hair contains the pigment granules. They may either be found to be pheomelanin, which are black-brown pigments, or eumelanin, which are red-yellow pigments. And it's the combination of these pigments that give us the colors of hair that we see. There may also be cortical fusi in the cortex of the hair. These are air pockets that develop in the shaft of the hair while it's growing. Also present at times may be ovoid bodies, which are a lot larger pigment structures that look like small little footballs in shape. The medulla is the central core of the hair. Medullas can take on a wide variety of patterns and distributions. As you can see from the examples on the left-hand side of this slide, they can be in pattern anywhere from uniserial to vacuolated to lattice to amorphous. The amorphous pattern is what is found in human hairs, while a lot of the other patterns, uniserial, multiserial, vacuolated, or lattice, might be found in animal hairs. The distribution of the medulla can also come in several forms, where it may be visible by just a trace of medulla, or it may be continuous. The medulla can also be dark in nature, or it can be clear in nature, depending on if that central core is air-filled or liquid-filled. If it's air-filled, the medulla will take on a dark appearance. If it's liquid-filled, it will appear more clear. The analyst will deal with both animal hair and human hair. Animal hair may be found naturally on a lot of clothing and textile items, and it can also be found by the animals themselves transferring hairs to the items or to vehicles. Human hair may also be found naturally shed on a lot of clothing or textile items, or it may also be shed through hard contact with particular items, such as what's shown with the windshield containing the hair fragment on the right-hand side of this slide. In forensic examinations, hair will help to establish contact between two persons, between a person and an object, or between a person and a scene. Depending on the amount of the sample, the hair is going to be analyzed for its various microscopic characteristics. First of all, the analyst will try to determine if the hair is animal or if it is human. You can see by the two hairs that are shown on this slide, there is very distinct differences between animal hair, which is the hair on the top, and human hair, which is the hair on the bottom of this slide. The microscopic differences are very easy to notice between animal hair and human hair. For color, an animal hair may change along the length of the shaft of the hair, so that the color that is found at the root of the hair may transition to a completely different color along the shaft of the hair to a completely different color along the tip end of the hair. Human hair tends to be more continuous in color unless someone has colored their hair artificially. The cuticle of the hair and the medulla of the hairs also have very different microscopic characteristics from animals and humans. And we discussed this previously with animal hairs taking on a wide variety of patterns, both for the scales that make up the cuticle or the spacings within the medulla itself. Human hair will always have an imbricate cuticle and an amorphous medulla when it is present. The pigment distribution is also very evenly spaced in human hair. With animal, it may be oriented more towards the cuticle of the hair. The cortex diameter of animal and human hair also varies greatly. With human hair, the cortex is going to be the majority of the hair itself in diameter. With animal hair, at times the cortex can be very small, with the medulla taking up a lot more of the space of the hair shaft. The root shape for human hair is generally bulbous, and several animals have very distinct root shapes. For instance, cat hair has a root that tends to appear more of a paintbrush shape, where dog hair tends to be more spade-shaped. Animal hair can be divided into 3 different types of hair, 2 of which are more commonly seen, either the guard hair or fur hair. The guard hair will tend to be a lot thicker and stiffer, and the overall shape of the hair shaft is going to appear to be a lot more shield-like, getting a lot wider in the center of the hair. The fur hair will be very thin and extremely soft. On occasions, they might also examine whisker hairs. For animal hair, the information received from an examination is a general species determination. So if we're looking at animal hair, we would be determining, is that animal hair dog hair, or is it cat hair, or is it human hair? With finding that it's human hair, you would go on and look at different microscopic characteristics. If it's animal hair, we would look specifically for the differences between different species. So determining things like the root shape or the various patterns of the cuticle or the medulla will make it easier to distinguish cat hair from dog hair, from horse hair, from fox hair, from coyote hair, and other species. Within the dog species, a forensic examiner is not able, from microscopic examinations, to specifically identify whether that dog hair comes from a German Shepherd or from a Cocker Spaniel. So we limit it to the species determination only. In some instances, it may be necessary to do color matching. The photos on this slide show a cat hair where you can see the paintbrush root. On the right-hand side of the slide, the top photo is of a fox hair, which has a vacuolated medulla that also displays the clear or light sections and also the dark sections that can be seen with medullas. The middle photo is one of deer hair, and you're primarily seeing the medulla of the hair, which is a lattice pattern. The bottom hair is a rabbit hair with a uniserial medulla. With human hair, the analyst is going to initially try to determine any racial characteristics for a hair, and then they will work with the microscopic characteristics to determine the portion of the body that the hair may have come from, and then other microscopic characteristics to make that hair a little bit more unique. They might be dealing with one of 3 different types of human hair. Languno hair is hair that is typically shed prior to birth, so it would be very rare to see Languno hair. At times, vellus hair might be examined in a laboratory. This is the hair that is extremely fine and very soft and present on newborns. Terminal hair is the hair that are typically found in most forensic examinations. So if we initially start our examination of a human hair, we're going to try to attempt to determine any racial characteristics for the hair itself. You can see from these 3 examples, the pigment distribution is going to be slightly different if the hair is from a Caucasian, or at times referred to also as European, or from Mongoloid, which at times is also referred to as Asian orientations, or Negroid African American. The pigments also vary in their distribution. The curling of the shaft will also vary, but in addition to this, make note of the cross-sectional shapes of the hairs themselves. The Mongoloid or Asian hair tends to be very round, where Caucasian or European is going to be slightly more oval in shape. And your Negroid or African American hair is going to be a lot more of an oblong shape, giving it its more curl to the hair shaft itself. Once determining any possible racial characteristics, the analyst will then try to determine the portion of the body that a foreign hair may have originated from. The characteristics for the various portions of a body do display different properties, as you can see from these three hairs on this slide. The hair on the very left at the top is a head hair. The hair on the right at the top is a pubic hair. It displays pubic buckling, which is one of the characteristics that makes pubic hair very easy to identify. The hair at the bottom of the slide is a facial hair, and you can see the coloring on it is slightly different because the hairs tend to take on a triangular cross-section shape, and at times they may also have more medulla to the hair itself. The analyst will also look for other microscopic characteristics of the hair, and it might be simply looking at the tip ends to determine if they're cut, if the hairs have any damage to them, such as the insect bites in the bottom photo. After determining a lot of the microscopic characteristics of the hair itself, the analyst will usually compare any foreign hairs to known samples that are available. This is done by way of a comparison microscope so that the analyst can look at both samples at the same time. The various microscopic characteristics that they take into consideration when they do the comparisons are the colors of the hair, and this would be along the entire length of the shaft itself. They also take into consideration the overall length of the hair and the diameter of the hair, the appearance of the tip end and also the root, and then more specifically a lot of the characteristics found within the shaft. The characteristics that deal with the cuticle, with the cortex, and with the medulla. At times, artificial treatments may be taken into consideration. So these would be things such as, is there hairspray or hair gel that is apparent on the shaft of the hair? Some hairs may also have special characteristics, and some of the more common special characteristics are individuals that have various diseases that affect the hair shaft. In addition to microscopic examinations, hairs can also be examined for DNA. Remember from our DNA discussions, there are two different techniques that can be used to obtain a DNA profile. Both of these can be used on hair samples, either nuclear DNA or mitochondrial DNA. Depending on the hair itself, the analyst will choose to do either nuclear DNA or mitochondrial DNA. To develop nuclear DNA, it's necessary for the hair to be generally in the anagen phase. So this is when the hair is actively growing, and there's going to be a lot of cells in the hair root itself, and then also the possibility, since the anagen phase is the active growing stage, it might be more firmly attached to that dermal papilla, so there might be sheath material that was pulled out with the hair. This also provides a lot of cells for DNA extraction. Mitochondrial DNA is necessary to be performed when there isn't the sheath material that's present or a lot of active cells in that root portion of the hair. So generally this is going to be what the technique that is performed on telogen phase hairs. Most of the hairs that are examined in forensic examinations are going to be telogen phase hairs, just because these are the hairs that are the easiest shed. Hair evidence can be recovered from a wide variety of crime scenes. Remember, once again, the average person will lose approximately 100 hairs a day. So even in normal, everyday activity, a lot of hairs will be shed. In a lot of crimes, there's going to be violent contact, which might also allow additional hairs to be shed. Usually hair evidence is detected by a good visual search. You always want to remember to use the most direct but the least intrusive method of collection when you're collecting hair samples. For foreign hair samples, these can be collected in several different ways, either through picking with tweezers, scraping items down, taping, vacuuming, or combing areas. One thing to keep in mind with hair examinations, it's always necessary to have a known reference sample if you're going to compare it back to individuals. Known reference samples are usually between 30 to 50 hairs. The reason for this is that every hair on your head is not going to be the same. As you can see from looking at someone, they might have hairs that have a slight, yellow tint to them or a reddish tint. They might have graying hair. So it's necessary to get a very good representative sample from the individual. It's also necessary to get hairs in several of the growth phases. So it's usually recommended to do combings and also pullings to make sure that you get some of the anagen hairs and also catagen and telogen hairs. Picking hairs with tweezers is very good in most situations because you're going to specifically know where those foreign hairs were collected from. As long as you're cleaning the tweezers in between exhibits, there isn't the concern for contamination. However, in some instances, this can be extremely time-consuming. So in some instances, items may be scraped. In instances where a ski mask has been used for a robbery, it might be hard to locate all of the hairs that might be contained in that knitting material if the individual has extremely short hair. So in situations such as this, you can scrape down the item to help recover any hairs that might be lodged in the weave of those fibers themselves. This can also be a little bit time consuming, and depending on the scraping methods or how much you scrape, you can somewhat damage hair. Taping can also be done, and it's probably a good method to use for very large areas. Because it does cut down a little bit on the time for the collections. However, with taping, then the analyst has to deal with getting the hairs away from the adhesive. So this can possibly damage the hairs prior to examination. And the same for vacuuming. It's very good for large areas. It is very quick. But the problem with vacuuming is that it's going to be a little bit too efficient. It's going to also collect hairs and debris from things that have been deposited well before the crime may have occurred. You won't know a specific location, and once again, through the vacuuming motion, you might have damage to the hairs themselves. Combing can be used for your known samples, but it can also be used to collect some foreign samples. One of the samples collected in the assault kits would be pubic combings, so this might be used to efficiently recover foreign hairs. And then pulling hair is specifically used for known samples. So this will allow you to obtain samples that are specifically in all of the growth phases. You need to keep in mind special concerns for packaging and storage of hair. A wide variety of packaging materials can be used for hair collection. You just have to keep in mind that hairs are very small items and can easily work themselves out of envelopes if they're not sufficiently sealed. Many laboratories will provide kits that have all the various containers in them, so that makes hair collection very easy. You also need to keep in mind if that hair is lodged in another item. If you do not collect the hair from the item, you need to protect it so that it won't be lost in the transportation to the laboratory itself. So if you can visibly see hairs that have become embedded in a windshield, if you don't want to collect the hair at that point, you need to somehow protect that hair so that the windshield can be transported to the laboratory. Like I said, most laboratories use kits and have packaging in the kits that is specific for hair collection for more known samples. Some of these envelopes can be used also for foreign samples. They would just need to be relabeled. And once again, always remember to seal your containers, whether it is an envelope or Ziploc bags. This provides an extra measure of security, especially if you put your initials across that seal. The microscopic examination of hairs and also DNA analysis of hairs have been accepted in courts for many years. The procedures for both of these examinations have passed Frye and Daubert hearings. When animal hair is analyzed, the examiner usually reaches a result. They will determine if it is animal hair, and then they will say what exhibit it was found on, and that it is consistent with a particular species. When microscopic hair examinations are performed, the analyst usually reaches one of two results. Either that the head or pubic hairs had characteristics different than the known samples, or that they were similar to the known samples. The statements will usually always contain the body part that the hair has been associated with, and if possible, also the racial characteristics. So using the terminology either Caucasian, Mongoloid, Negroid, which is found in most of the literature when we talk about hair examinations, or European, Asian, or African. In some situations, it might be possible that the hairs don't display enough characteristics for identifications. So in some situations, there may be statements of the hair examinations being inconclusive, or that the samples have insufficient characteristics. When DNA extractions are done on a hair sample, the examiner can usually reach one of two results. They will first usually say whether or not the hair is from a male or a female, and then if the profile matches an exhibit, they will state that, and then also give a frequency of that profile based on the U.S. population. There's also circumstances where that profile might not match. In some situations, the extraction of DNA for hair may be attempted, but it may result with inconclusive results.